All right, I want to welcome everybody to our webinar this evening. We had a little bit of technical difficulty behind the scenes, so I apologize for the delay in getting started. Thank you to everybody who sort of hung on there. We're really excited to be here today. I'm going to be presenting along with Dan Fowler, who's the Chief Medical Officer for Rapa Therapeutics, about the RAPA-501 EAP for ALS. Next slide, Catherine. Great. Um, and so the other person at the Rapa Therapeutics company that we want to acknowledge is Brian Radecki, who is the CEO of the company and really committed to this program, really committed to developing RAPA-501 for people with ALS and has been an incredible partner to Dan. And I've had the pleasure of working alongside them on this EAP, but also on the clinical development program. Next slide. So I want to take a few minutes and just explain sort of what RAPA-501 is. These are T stem cell infusions, and they're autologous, and we're doing this in people living with ALS. So there's a process for making and infusing these cells. Autologous means they come from the person who will be infused. So they're coming from the person with ALS who enters the trial. The first step to all of this is that people enter the trial as participants, and their T cells are harvested through a process called apheresis. Now, we don't just take off T cells. This apheresis consists of a blood draw that goes on for a number of hours. The blood goes out through an IV to a machine that holds on to just the white blood cells. The white blood cells are the ones that fight infection. Now, it doesn't keep enough of the white blood cells to make people immune suppressed, just enough that we can make the product. So those white cells, whatever they may be, all these different white cells are collected through this very standard clinical process called apheresis. And then those cells are shipped down to the Rapa Therapeutics manufacturing facility. And there, the T cells, all T cells, are isolated. Now, I want to be clear, it's not the regulatory T cells that are isolated, it's all T cells that are isolated. And they go through a process called dedifferentiation. That means all the T cells become sort of a deprogrammed, kind of very naive kind of T cell. And they're all sort of similar at that point. And that's done through epigenetic reprogramming. So there's no sort of genetic manipulation that's gone on, and that's Rapa's therapeutic process for the cells that they've put together based on many years of research. And then there's what's called redifferentiation. That is, we make all of these T cells, different kinds of T cells, helper T cells, killer T cells, regulatory T cells. We make them all into sort of naive T cells, and then they go through a process to make them back into a kind of a regulatory T cell, but it's a very naive kind of a regulatory T cell, and it shares some properties with other kinds of T cells, effector T cells, to make it a very resilient T cell. So they're grown in a way that make them want to stay as these kind of naive regulatory T cells, and that's different than just having regulatory T cells, which can switch into killer T cells or helper T cells when they're infused back into the patient. So once those T cells have gone through dedifferentiation, redifferentiation, they're then parsed out into doses, and they're frozen, and they're shipped back to the site. And then the site takes those frozen cells, and when the participant is there in the infusion center, they thaw the cells, reawaken them, and they're put back into the body, and they act as anti-inflammatory cells. Next slide. So I've talked about some of this, but this is a little bit of an overview of sort of how that process of dedifferentiation, redifferentiation sort of happens. And it has to do with blocking the mTORC1 and 2 pathways, selecting cells that are metabolically most fit. This is sort of allows us to have the strongest T cells that go back into the body. There is a process of removing checkpoints and making these T cells into sort of kind of naive, we call them T stem memory cells, This gives them a long-lasting property that's different than just Tregs. This is the step that makes them more likely to survive and less likely to convert back into some other kind of T cell. And then there are certain cell surface markers that then begin to appear that make them particularly anti-inflammatory and good at reconstituting the immune system in a positive and healthy way. And that is the final step in making these RAPA-501 kind of cells their final state. So we often shorthand this to sort of a Treg program. It's not really just Tregs that are going back into the body. It's these sort of highly engineered RAPA-501 cells. Next slide. So with that in mind, we want to just update you on status, and we make these slides, and then we continue to progress. And actually, this slide is just a little bit out of date, and I'll tell you how at the very bottom. But we screened 62 participants. There were a few people who screen failed. 57 of those people enrolled in the study. They underwent apheresis. There were 2 that left the study prior to apheresis and never had the apheresis because of disease progression. We're targeting people who are later in the disease, and that it became apparent that the protocol was too onerous, unfortunately, for 2 participants. And then there were 55 who went on to have the apheresis. Of those, 3 did not actually get the drug in a way that would make them evaluable. We wouldn't be able to sort of evaluate the effect. They didn't have the cell infusion or didn't get enough of it for it to be evaluable. And then there are, at the time of the slide, 49 who had at least one of their RAPA-501 infusions. I think that's actually all 51 now have had one. We may still be waiting for one to have an infusion. But all of the aphereses are done, and this is more than our original goal. We originally got a grant to do 40 participants. We got an expansion grant to go up to 50, and then we expanded beyond that by finding some efficiencies in the budget. Next slide. And Dan, I'll hand over after this slide, but we wanted to acknowledge the 11 really excellent centers who have put together their expertise in ALS, along with the expertise in the hematology units in their hospitals and their institutions, to really put this novel cell therapy in action. And it really is bringing together a number of different sites, or pardon me, different teams within the site to really approach this very novel therapy. Next slide. So Dan, do you want to talk from here?
Yeah, thank you, James. I'm Dan Fowler from Rapa Therapeutics. Pleasure to be here today. As individuals on the call may know, the Expanded Access, the Act for ALS, has 2 main objectives that we brought to bear when we wrote this protocol and implemented it. Number 1 is access. So we wanted to treat individuals who were not eligible for other trials, including the conventional RAPA-501 trial in earlier stage patients. We define that by individuals with pulmonary vital capacity of less than 50% of normal, predicted normal, so a very advanced respiratory situation. The other main objective for the EAP coming out of NIH is there must be high-level research, intensive research to go along with extending the treatments to patients. And as James says, this is an immunotherapy. So our main goals were on the immune system to understand what's going on in these advanced ALS patients, and also are we able to move the needle by putting in these RAPA cells. The next slide. This is just a more detail of what James talked about, that the patients come in for screening on the left. We do an apheresis procedure to collect some of their immune system. We manufacture those cells in the incubator. They're frozen. Then they get shipped back to the treatment site, the 11 sites that James mentioned that we work with. These cells are shipped to those sites and then thawed and infused on 4 separate occasions, each infusion separated by about 6 weeks. And then the patient goes on to have follow-up visits, and we measure many of the key outcomes that are classical for ALS studies. And in addition, we added in a lot of research studies and remote monitoring through Everything ALS organization. Next slide. This is an example, I believe, is one of James's patients who on the left came onto the study with low slow vital capacity, about 45%. And it's predicted that individuals will have a continued decrease in these values over time. The x-axis here is months on study. And in this patient, the breathing test went up and kind of stayed at the same level or slightly higher through the duration of the study. And we've seen this pattern on many of the recipients. Obviously, it's still early days of evaluating all of the data, but we're cautiously optimistic that we're preserving some pulmonary function. But this is one of our key metrics for the study. Next slide. We don't have a lot of time today to go over, so many immunology data and so forth, but one of the purposes of this study, one of the objectives was to develop with Origin Data Sciences and AI-driven, so prognostication of survival. So we know in ALS that there's some heterogeneity, even in patients with advanced stage disease. Some are predicted to live a little shorter than others. Some may be a slow progressor. So we developed a model with Origin where we then, in this slide, our most recent cut of data was with 34 patients where the model predicted survival. And then what's plotted here is the actual survival on the top, which is the RAPA-501 recipients. And you can see it's early days. However, cautiously optimistic that the survival is improved relative to the AI-generated prediction, the virtual controls. We also have been looking into other databases, so-called real-world data or data of other patients that are not on our trial, and we collected 650 and more individual survival data. This is a group of patients that's in the PROACT database who we selected them on the basis of them having less than 50% breathing capacity as well. There's also a lot of caveats to this. Obviously, it's not a control group within your own study, but it's the best we can do. And a key thing going forward is to further improve these models and to look at this information in the full set of all the patients we treated. The data will mature over the next 6 to 12 months. Next slide. So the last slide, I believe here is the next steps. We call this the RAPA-501 EAP 1.0. And the goal is to finish all of the infusions, as James mentioned. All of the patients that are allocated to the protocol have been accrued, so we don't have any further screening slots, unfortunately. So the goal now over the next year is to finish up all of these infusions. 1 or 2 patients are still waiting to get their first infusion. That'll happen in the next week or 2. And then we have to analyze all of the data and pull the whole picture together. In the meantime, down the middle, as you probably know, we're hopeful that there will be another grant mechanism for funding, and we are intending to make an application to the NIH for a continuation of this program. We'll call that the EAP 2.0 if we're optimistic about that, but that's our intent. And the data that we have generated will be very useful in trying to make a compelling case that this program should be continued. And then on the left, the important point here is we've learned so much about this subset of individuals, and we're cautiously optimistic that this should actually be a part of our product development cascade, where instead most companies focus on the early stage of disease. We're very interested in working with the regulatory bodies to try to evaluate RAPA in the advanced stage of ALS. And then I think that was the next slide is maybe opening up to question and answers. Thank you all for your attention.
Yeah, thanks so much, Dan. So I think what we'll do is take a few questions. It's always fun to talk about this with you and sort of, go over it soup to nuts, what's the process for making the cells, and then, how do we think they might help, and then finally, what's the design of the CAP? And I just would underline that, the EAP is designed with these dual missions in mind, and we really are learning a lot about, how you run an EAP, what are the features that are going to make it successful, and then we look forward to so much of the biological data that will come out of this, as well as all the clinical data. Okay, so we'll turn our attention to some of the questions. And I don't know that we'll be able to answer all of these, but we can certainly go back to our data and know that people will want the answers to these. So one is, what percentage of participants had sporadic versus familial ALS and slow versus fast progressors? And that's a really good question. I don't know that we have that tabulated right now.
Yeah, James, I believe the hereditary form is somewhat less than 10%. We've had some, but it would be in the 5 to 10% range.
Which is what we'd expect.
Yeah, and is in terms of slow progressor phenotype, when we look at the origin data on the virtual control, because that predicts their actual survival, I would estimate that 25 to 33% of the patients would fit into the slow progressor phenotype.
That's great. And one of the things that becomes a little bit challenging in an EAP is it's different than an observational study. So we can't actually look at what happened and say who is slow or fast, because we might be doing something to make people into slow progressors. In other words, if it was an observational study, you'd just look and say, well, here's how many percentage of the people in our study went slowly. They are the slow progressors. And here's the number that went fast, and they're the fast progressors. Here, it may be that those percentages are altered. We hope that those percentages are altered by the treatment that everybody is receiving. Okay. And let's see. There's a question, when do you expect EAP2 to start enrolling? And I guess I would start the answer to that by just saying that this is, EAP2 is, what we want to do is propose that in a grant, and then hopefully we could have funding for it, and then hopefully quickly hit the ground running. These grants are usually funded in the autumn. At least historically, that's how it's been. We don't have a formal RFA or request for applications that we can look at and say exactly what their timeline is. Dan, did you want to add anything to that?
Yeah, I would just say we would be cautiously optimistic that the proposal that NIH would release a application in the next short time, and then there's competition, there's other people applying, and then there's 6 to 12 months would be an optimistic if we get the funding and can get back on track, I would say.
This is here's a question, Dan. Will patients be tracked past the end of the study in order to understand durability of the effect? And is there any prior data or expectations about this?
It's a good question. We follow survival on the protocol. We put in an amendment so that survival of the individuals can be followed. So we're going to make a great attempt at that. But the grant has a pretty strict 3 year cutoff time. We'd like to follow longer, but we're going to be running right up against the NIH milestone in terms of that. The question about the durability is a very good question. I believe that there should be good durability. We've had some of the early patients that had some breathing improvement. They seem to be stable over time from communications that we've had with the investigators. The immune system, as you know, when you get a vaccine, it can last a long time, even the lifetime of an individual. And we engineer these cells, as James was saying, to have some stem cell properties. A stem cell can divide in a very durable manner. So we're hopeful that there will be a durable effect, but more studies will need to be done to actually prove that about the optimal timing and duration of therapy.
Yeah, that's great. It really be exciting to see this as the clinical development program goes on. Just for sort of everybody in terms of definitions, we sometimes talk about clinical development program, and that really encompasses all of the trials and the EAPs that are done under one program to guide that program toward learning more about it and also toward, hopefully, with success, eventually regulatory approval. So it's kind of that family of EAPs, trials, phase one, phase two, phase three, that hang together. So we look forward to continuing the EAP and as well as the clinical development program. So there's one question: why were some participants chosen for round two to sort of have a round two and some not? And I'll take a first crack at that. We did want to have when we expanded the project, we saw an opportunity to see how people would do if they had more extended therapy. The people who were selected, it was a lot of it was simply timing. People who were ending this their first round of the protocol in the window that we only had a few months to enroll for the expansion phase. So it was people who were ending and then were able to kind of continue on because of the timing of their completing round one of the EAP. And that was really the main determining factor.
Yeah, I would just add to that, James, the retreatment was made possible by this supplemental funding, which I believe was about 12 to 15 patients, and only a certain part of that, which was 3 patients, could be included in this retreatment cohort. And that's what you usually see in a safety feasibility study is this even possible? What are the side effects and so forth? So that's why we were somewhat limited to only 3 patients, and those were the first 3 patients that had came off and were referred back to the study. I personally believe it's a very important topic of retreatment. I think patients that are thought to maybe get benefit should ethically, every attempt should be made for continued therapy with the caveats we don't know about the durability and the timing. But it's definitely something that we're committed to including in this new application, for example.
There are a number of questions that come together, Dan, to really that ask about the clinical development program. What has been the effect of this EAP? Has it had an effect on your thinking about the overall clinical development program? And if so, what is that effect, and where do you see the clinical development program going for people who, sort of fit the parameters of an EAP, but also people who are earlier in the disease? Kind of a broad question.
Yeah, it gets into a lot of different issues, right? The EAP really woke us up to the fact that the immune system in these individuals, that's part of what we're studying, is very dysfunctional, perhaps even more so than earlier stage patients. So if you're doing an immune therapy like RAPA-501 and you're trying to reconstitute the immune system and provide more balance, it's probably the patient group that where you might see a signal of efficacy easier than if you're going into the early diagnosis. The early diagnosis is also complicated by the fact that in a lot of research, you're looking for a survival advantage. And in cell therapy, that's very important because we're kind of limited in the scope of our research. To do 50 patients or 100 patients on a more definitive trial is a very labor intensive and costly maneuver. Whereas if you're doing an infusion or a tablet, you know, in ALS studies, sometimes the patient numbers can get up into the a couple hundreds. So you really need to see a very large effect size when you're doing cell therapy. And I believe that the advanced stage ALS is probably the right place for us to be. That's not to say that we're not interested in moving it up earlier. Obviously, if something's working, you want to get it to individuals before more quality of life is compromised.
Yeah. And there are some people who are sort of asking about, future plans for placebo group, for larger studies, for studies in people who are earlier versus later. I wonder if you could just comment on those things and what your process will be for those.
Yeah, the control group is important. I was impressed with our collaboration with Origin Data Sciences on this AI modeling, and I believe that is the future. Now there's a gap between what we believe and what researchers believe, and maybe what is accepted by other individuals in regulatory positions and so forth that have maybe a more conservative view of things. So, and we're meeting with regulatory individuals all the time and going through these aspects. We're certainly willing to consider randomized controlled studies. We understand that's the gold standard for all therapies. But in ALS, particularly advanced stage ALS, it's a very complicated situation because there are no real therapeutic options, and we're very cognizant of that when we make these decisions. So it's an evolving landscape, James, and as you know, and we'll do our best to navigate it and try to get the funding to support all of these issues or all of these landscapes, whether you're talking advanced stage or moving up to earlier interventions.
There's one question I'll just take. The question is the ultimate goal to slow down progression or, have people regain function? And I think that's probably a spectrum. I think if we could slow progression enough, then it might give the body a chance to recover some of to sort of reinnervate some of the muscle with existing motor neurons. So it may be a matter of degree of slowing. When we power trials, we're often thinking about slowing and how would we detect it if a drug or an intervention, in this case, cell intervention, was powerful enough to halt progression or reverse progression. We think our trial design would pick that up. What we want to do is think about how many people do we need to test this in order to detect a small but meaningful slowing of progression. And that's why you hear us talk about slowing of progression as kind of how we've designed the trial. It's because we want to know what's the smallest meaningful effect that we could pick up, and anything above that we would also be able to pick up. So that's kind of why we talk about that. There's a question about how many of the participants were bulbar onset. And I don't know that offhand. Dan, you might have that number.
Yeah, it's about at least a third, I believe, somewhere in that range.
I think that's right. I mean, again, what we see for sporadic versus familial, for bulbar versus limb, they follow kind of what we know about the disease. I think male and female is roughly like that too.
Correct.
Treg levels before and after therapy, what's the plan for looking at those, and how will they be important? And what are the nuances of these not being exactly Tregs?
Yeah. Yeah, the nuances are these things are very, in my opinion, very difficult to define You need to, and also the T-reg numbers are just a small part of the puzzle, I believe. The There's a lot in immune dysfunction, right? ALS starts as an inflammatory disease. So the immune system is not supposed to be in the brain agitated by degenerating tissue, but it does get that way in ALS the more we learn about different models. And then when the immune system gets activated to the tissue damage, there there's a hyperinflammatory stage where you see excessive inflammation. And then we know from many other diseases, then it becomes more complicated after that, where you can actually see some immune suppression. And then individuals are not able to respond to normal things like viruses and bacteria. So that's an open question, James, is to what degree does ALS resemble some of the other diseases, the autoimmune diseases like lupus or MS? And I'm on the side where it resembles them. After doing this study with an open mind and doing all these measurements, I believe it really does reflect a little more on the autoimmune side than a pure degenerative condition with a little involvement of the immune system. So I think it's part and parcel. And measuring Treg cells, it hasn't been too fruitful for us. We do measure them, but we like to focus on the more global aspects, like what is the vast repertoire of all the T cells, the clonality, and what are the immunosuppressive or inflammatory markers. So more of the downstream events where kind of the loss of Treg maybe led to these events, but it's hard to find that signal. The one of the reasons is that we draw blood to measure these things, but a lot of the action is going on in the tissue.
Yeah.
So you're sampling the blood and you're trying to make these conclusions, but it's tricky business because a lot of the action is really going on in the tissue that which we are not able to sample.
Yeah, yeah, that's right. I sometimes think when I think about all the complexities of the immune system that I went into neurology because it was relatively straightforward. Um, clearly that's not true, but boy, there's a lot of complexity in the immune system. We
Yep.
have more questions here. I want to be careful about time. We did get started a little bit late. We've run a little bit late. We have a few more questions, and we'll try to get back to those people who asked the questions. I guess I'll just say quickly there are a couple of questions about people who've been exposed to RAPA 501 either in the EAP or in the trial, and is it possible that they would have access to an EAP in the future? And I guess I'll say that um, we modeling this off the previous EAP, it was okay for people to have had the RAPA 501 treatment in the past. There wasn't like an exclusion that said if you've been exposed, you can't be a part of the EAP in the future. So that's important. There was a question about whether we involved people who had C9orf72, and the answer is it wasn't. We didn't specifically enroll people with C9, but they certainly were a part of the project and will continue to be in the future. And then I think with that, we should probably pause. And Dan, thank you for being such a partner in this and just a stalwart in the world of ALS, really moving this program forward meticulously, looking for the biomarkers, looking for the clinical effect, thinking about what the clinical development program ought to be. And
Thank you, James. And thanks everyone at MGH. It's been a fabulous collaboration. And thanks particularly to all the patients and caregivers who are the real champions here of needing investigational therapies and improving the research. And I think this EAP was very enlightening to us, and we hope to keep learning more.
Great, thank you. And thanks to all the site investigators as well who've really.
That's true too. It's been fabulous.
All right, thanks so much.